Hey everybody, I'm Brenna Fox, and for the past year, I've been working on a protocol that allows for same-day direct-from-plaque phage DNA sequencing. Sequencing phage DNA is a key step to gaining insight on the genomic makeup and life cycle of new phages. This sequencing allows for identification of viral components that have potential applications in biotechnology. Now, despite relying on DNA sequencing for phage characterization, traditional protocols for isolation and sequencing have significant limitations, including difficulties analyzing a mixed population or rapidly identifying contamination in a sample. Currently, the standard method of sequencing phages is preceded by the production of highly concentrated DNA and sequenced through Illumina. This protocol relies on a series of plaque isolations and amplifications to produce purified lysate and subsequently extracted DNA. Due to the intermediate steps of purification and amplification, this protocol often takes up to 2 days, and the requirement for DNA extraction further adds to the cost of each sequenced sample. The new method that we have optimized relies on the input of only one plaque, which can then be directly amplified using PCR rather than through additional plaque assays. Since the amplification and DNA extraction are bypassed, the full protocol can be performed in less than 5 hours, allowing for same-day sequencing readouts. Additionally, the use of nanopore technologies allows for easy and lower-cost in-house sequencing. This protocol is composed of 4 primary steps. Plaques are initially isolated by soft agar punches and resuspended in TE buffer. Following isolation and particle lysis, the phage resuspension is fragmented by a transposase complex, which simultaneously inserts known regions into the DNA. This transposase cleaves and inserts randomly, which allows for targeting by PCR. This known region is used in the subsequent PCR step, as the barcoded primers will bind to the insertion. When PCR has concluded, all samples will be both amplified and uniquely barcoded. During library preparation, all samples can be pulled together before adding an adapter and sequencing. So this diagram contains a visual representation of each key component that's added to the phage DNA. All 3 of these reagents are found in the Nanopore Rapid PCR barcoding kit, which contains enough reagents to allow for up to 24 samples processed simultaneously. However, in-house production of primers could allow for scaling the number of samples with no upper limit. During fragmentation, a transposase randomly cleaves the DNA and inserts a known recognition sequence. Following the fragmentation, barcodes are added to the DNA using primers which bind to the 5' end of the recognition sequence. Right before sequencing, an adapter is added to the pooled samples, which bind to the purified DNA and allow for attachment to the nanopore flow cells, therefore allowing for base calling. The traditional method of amplification with plaque assays, followed by DNA extraction, is time-consuming and costly for each sample processed. We tested this alternative method using PCR for amplification rather than plaque assays, and sequencing using nanopore, which allows for both a decrease in time and cost. The optimized protocol was tested on 6 previously Illumina sequenced phages. Comparing the data from both the published genomes and the protocol's output, it was found that the mean average nucleotide identity of all samples was 99.97%. Additionally, out of the 6 phages, 4 of them had an exact 100% ANI. For the one phage with a slightly lower ANI, the sample has been repeatedly passaged since its Illumina sequencing 8 years ago, so it is possible that the ANI would be 100% with an updated Illumina run. This adapted Nanopore Rapid PCR protocol demonstrates nearly a 100% sequence similarity with the traditional method of DNA extraction and Illumina sequencing, and only costs $7 to $15 per sample. This high accuracy, in combination with up to an 89% decrease in time required to process samples, makes this new method a rapid alternative for high-throughput sequencing that can be performed in-house with same-day readouts. As there is no requirement for a pure culture, this protocol can be used to quickly identify contaminants on a plaque assay or analyze the diversity in a mixed sample by sequencing multiple plaques from the same plate. If you would like to know more information on the technical details or protocol validation that we performed, I have included a link to the bioRxives text. This research was done under supervision by Dr. Patrick Lipachevsky, and all phage samples used in this research were isolated by the McGill C-phages student cohorts of 2022 and 2023. I would also like to mention that this protocol was recently used by Dr. Adam Rudner's lab at the University of Ottawa, and his lab will be presenting the research shortly. Thank you so much.